again welcome.
I request all the dignitaries to please come on to the dais. May I request our principal, Dr. R.V. Patil sir, to felicitate our today's guest, Dr. Ramdas Chute sir. Thank you, sir. Now I will request Dr. Nisha Rani Randpise ma'am to felicitate Dr. Gayatri Deshmukh ma'am. I will request Mr. Prashant Mai sir to felicitate Miss Priyanka Kumbar ma'am. Thank you, sir. Now I request Dr. Praveen Bade sir to give preamble about the program. Please, sir.
Good afternoon, all. Respected guests who have joined offline and also online. Good afternoon, Dr. Raju sir and Dr. Lakshmi ma'am. So today we actually gathered here on World Hepatitis Day to inaugurate our Center for Drug Discovery and Development. I actually thanks all the guests. Actually, it was very short notice, and they have actually taken time out from their busy schedule, and they have come here today to inaugurate our Center for Drug Discovery and Development Center. Actually, uh, we are doing the research here. All the departments, they are already doing the research, and there is research already going on. But the purpose of this center is to have an interdisciplinary collaboration which can help us to take basic research to the clinical level. So like taking genomic to the clinic. So that's the motto behind this center. So I will just give you a brief introduction, the layout of today's program. So we are going to start with opening remark and uh, basic welcome from our principal sir. And after that, we are going to have a information about the facilities which we have got here at SEOP and how we are going to utilize it in our new Center for Drug Discovery and Development. That we are going to start. And after that, there will be a um, short informative session from our guest. So we are going to request our guest to have some um, their view regarding the center after having our presentation. So that is going to happen. And after that, we are going to have vote of thanks. So that's the summary from my side. And we are going to start. So I will give back to Shinda madam.
Thank you, sir. Now I request our principal, Dr. R.V. Patil, sir, to give opening remark and welcome address.
Respected chief guest of today's program, Dr. Ramdas Kute sir, uh, Gayatri madam and her colleague from Aptech Foundation, Aptech Research Foundation, my dear faculty members, students, and the online participants who agreed to our invitation and joined. Welcome you all for the inauguration of Center for Drug Discovery and Development. At the outset, I welcome our chief guest, Dr. Ramdas Kute sir, who is heading one of the leading pharmaceutical company in and around Pune. You'll come to know through his uh, talk that what facilities his company is having there. I will just share the genesis of this Center for Drug Discovery and Development. You will know that Bade sir joined our institute. I will just take a little more time because
Okay.
okay. Laptop voice. Okay. someone just confirm whether our online participants are listening to us. So it should not happen that we are talking here and the people who are very far from us, they're not listening to us. It should not happen. Okay, so I just requested, I will take little more time to have my insights on the genesis, how this idea started. I don't know about the outcome of this idea, but all of you know that Dr. Bade joined us in the month of January. And before joining, he shared with me some of his ideas. And one of the idea which clicked me that he wanted to have such center where interdisciplinary research will take place. And he shared how to go ahead with such kind of center. We, as a teacher, we know that our expert areas are well defined and we just work in our areas. But if we just enhance a little of our skills and if we collaborate with our own colleagues, we can do fantastic research. So with that thing in mind, I give entire responsibility to Dr. Bade, and it is really commendable that he himself took a lead, though a leader was there on second floor, but he himself took a lead. He arranged so many discussions. He arranged so many meetings with all the faculty members, and he came up with this idea. I'm happy that my faculty members can do such things. And it's not just praising you, but this is what I see at the as an outcome. This is the genesis of our Center for Drug Discovery and Development. In one sentence, I say that this center is built on our team. Because of only team, it was possible. Otherwise, such things are not possible. Through this center, through this CenDDD, we have planned collaborative research and collaborative efforts. We don't know our strengths. If I share my strengths, I'm not boasting myself, but I have been there in research and my basic research, as Badesar told, and I have so many collaborations, not myself, but so many of all the faculty, almost you are having your own collaborations. Make use of those collaborations, bring them together, we can do fantastic things. I'm sure with this intention, the center is going to have its success for sure. It is for sure. Even if I be your leader or not, I'm sure that this center is going to be successful. Only thing, you have to work in a team. I remember this CenDDD in our college, because of the prevalent situations and the circumstances, so we as a senior guard employees faced since last couple of or maybe more than that years. Some of the faculty members sarcastically used to say he's doing DDD. He's doing DDD sarcastically. And that DDD was meant for dedication, determination, and devotion. Despite any outcomes, why you are putting DDD? This was sarcastically said by many of my colleagues, and but today I'm feeling very happy that the same DDD we brought into reality. And this center for drug discovery and design, these DDDs will remain there. So dedication, determination, and devotion. For every success, I know that devotion is at most important. It always, devotion always brings a good results. So we should remain committed to whatever we are expert in. A good devotion will lead us to the determination, and doing a few things consistently will surely prove our determination. And finally, with the determination, you know that dedication comes, and dedication is always responsible for getting or having accomplishments, as you know. So with these three DDDs, which are there in our CenDDD, we are for sure we will fetch a success. I, through my studies and through my observations since last 1 year or so, many of our faculties are experts in many areas, and they themselves, they don't know. So, for example, I've just listed out a few of the expert areas where we can work together. In our discussion, sir just brought in a picture material science research, material science studies, or material synthesis. That is one of the area where some of the faculty members are expert in. They can explore that area. Through my research areas, I came across agricultural sciences, agricultural research also, where you will not believe, but agricultural science also goes hand in hand with our pharmaceutical chemistry field or pharmaceutical sciences. They also require formulation. Do you know this? We are not exploring that area. Someday we will be there in agricultural sciences. We will be there developing some insecticides or pesticides or some such material. So let us go ahead, explore that area, agricultural sciences. I have already worked in such area where there are tremendous scope in working in that area, and agricultural people are not expert in these areas. They will not explore that area because of the limitation in their syllabus or curriculum. Let us give hand to them, and let us fulfill our dreams through the center. Most of us are afraid of the term genomics, next generation sequencing, and so on. Mali sir is expert in pharmacology, and he will surely have he will surely develop students and enhance his own team's understanding of genomics, next generation sequencing, and so many things are there. Today morning we arranged one session that hepatitis. I wanted to ask one question, but I felt that it should not be asked in front of students, and why there is no vaccine for hepatitis C. That question should have came from students. Why there is no vaccine for hepatitis C? There should be some efforts. But the biggest problem was almost there are 7 genotypes of hepatitis C, almost 90 subtypes of hepatitis C, and it is not possible to develop a single vaccine for all hepatitis subtypes. That we know, but there should be some attempts in that area also. Vaccination, vaccines, development of vaccines. Computationally also, vaccine development is possible. Let us explore that area. Some of you are experts. Some of you can gain expertise. Try to learn that. So that is the purpose of my CenDDD. I just enlisted one more area, crystallography. Some of the faculty, in fact, our Nisha madam is also expert in crystallography, co-crystals, and so many aspects are there. Crystallography, there are fantastic sessions or courses available for crystallography in NPTEL. Do attend that and gain expertise in crystallography. That will surely give us the fantastic results in pharmaceutical sciences, believe me. Obviously, our center's name is Drug Discovery and Development. Drug discovery, it begins with design itself. And you'll surprise that drug design itself, it is a so much huge area that in one's lifetime area, he cannot explore all those things. There is a limitation. We should give that understanding or give those assignments to our students. Only through students it is possible. So that drug design and drug discovery itself, it is a very vast area which we need to explore through this center. Let us extend our hand to all the collaborators. I surprisingly saw Dr. Rajiv Singla. I have been in contact with him since maybe, if he remembers, I have been in contact with him on ResearchGate since 2014 or so. And he himself is editor of one of the journal, and currently he is in China. He knows where drug discovery can lead us, and he is working in pharmacology and his expert areas. We are having that tremendous capability in drug discovery itself. In computational chemistry, I'll tell you, all the collaborators, all the participants online, that we are having a fantastic expertise in handling of HPC system, which can account for almost 200 um, uh, latest laptops together. If you put together around 200 laptops, whatever they can do computations, in a single minute, we can do it on supercomputers. What we call it as HPC clusters, high performance computing clusters. We are having access to that. I, being a SCOP part, I am having access to that. We can use it. We can use such fantastic systems. And who are not having that system, they can approach us, get use of us through this center, and they can produce their results. So those areas, including drug design, you go for docking or go for molecular dynamics, high level molecular dynamics, or any of other computational studies. That expertise we are having, and I'm slowly making people comfortable with these areas. And I'm sure in coming, I always believe in long-term planning. And I'm sure that if I consistently do few things, surely it will give some results to me. And some of the faculty will gain expertise in coming years, I'm sure. Maybe some of the students, I saw these final year students, some of the students are very good in arts. One of your friend, if you remember, he's fantastic in arts and he can prepare graphics and such things. Let us explore that area through programming or graphic designs and such things that we can do through this CenDDD. Translation research is there. Let us transform the traditional knowledge, Madik Madam, traditional knowledge into the preclinical candidate. Let us do it through our pharmacognosy department. So all these possibilities are there. I just wanted to share these thoughts with you that there are tremendous opportunities waiting for us through this center. I took little time. Actually, students are there. Students, they are obviously they will be a direct part of this center, CenDDD. Whichever student want to pursue his careers in research or in academics or pursuing higher education or whatever startups they want to go ahead with, or any of the ideas, bring those with us. These are my faculty. Discuss with discuss with them and let us utilize this CenDDD. With all these purposes, all these thoughts in my mind, and with my uh, faculty members like Bade sir and all the HODs and all faculties and some students, today I'm very happy that today is so productive for me, including some celebrations, so which was surprising to me. Uh, so with these thoughts, I think we'll go ahead with the rest of the program. Thank you. Thank you very much. Thank you.
Sir, thank you for your valuable words. Now I request all the dignitaries to please sit down. Now I request Dr. Praveen Padesh to give brief introduction about this Center for Drug Discovery, Development and Services. Please, sir.
So good afternoon all again. Actually, sir has already spoken uh, a complete introduction. So I am not going to actually talk about the center, but I just want to focus on the vision. So our vision is to actually give high quality R&D facility and also provide services. So that's our vision with this center. And what we are trying to do in collaboration with startups and companies, we are going to work together to bring different, like sir said, product from natural products and also from the synthetic one, because we have got a lot of nice expertise, I think, from all the areas, specifically from herbal technology is there, nanotechnology is there. What I think is that what we are missing here is only the preclinical studies and the animal studies in different areas. We got that. We got different departments where HODs are actually doing the research already on different types. But with the center, we are going to come together as an interdisciplinary team where we want to explore, different formulation prepared using nanotechnology or using herbal technology or using semi-synthetic formulations to move further. And while doing this, we are not actually just doing as part of the central drug discovery and development, but we want to actually bring the pharmacy student into it. So that way they are going to actually work on the live project, which are going to be useful for the companies. And also that skills are going to be beneficial for students when they are moving out in the field. And to do that, we are actually collaborating. We are making sure that we are having MOU with pharmaceutical companies, CROs, and different companies, different areas. So we are going to go from basic research till clinical research in collaboration with the pharmaceutical companies and academic. So that is the vision and mission behind the center. So now we got different areas which are going to be explained by the expert staff. So I'm just going to request now Dr. Sadhna madam to carry on the further presentation.
Thank you, sir. Good afternoon to one and all present here. And today, here is inaugural function of CenDDD. There is nothing but Center for Drug Discovery and Development. So I'm going to present over here which kind of facilities we can provide from Pharmaceutical Chemistry Department. So here, there are the thrust areas from pharmaceutical chemistry department, which includes the synthetic chemistry, then computational chemistry, molecular dynamics, 3D, 4D QSAR, and also binary QSAR, then pharmacophore-based modeling, and also we are going to provide drug design in detail. That is nothing but ML, that is nothing but machine learning and artificial intelligence-based drug design. Then homology modeling using modular 9.25 version. And then next one is the ADMET, that is nothing but ADMET, that is nothing but absorption, distribution, metabolism, excretion, and the toxicity predictions also with the help of software, and RDK database screening. So that synthetic chemistry in detail, that is nothing but we are going to synthesize different kinds of molecules with the help of various techniques. That includes the microwave assisted synthesis, then also synthesis with the use of homogeneous and heterogeneous catalyst, solvent-free reactions, also multi-component kind of reactions, and ionic liquid-based reaction. So after synthesizing different kinds of molecules, so we have to do the computational chemistry. So we are going to provide such a kind of computational chemistry things that is nothing but regarding with the drug design, which contains the, or with the help of softwares, that is nothing but Autodoc software, Autodoc Vina, and Doc6 software. So we have the facilities. That is nothing but we have the such a kind of softwares also. With the help of these softwares, we can investigate or we can discover new molecule, and after that we can do the preclinical clinical study also. Then also which includes the protein peptide docking, protein docking, then covalent docking, then enzyme docking also. And in case of the molecular dynamics, the simulations, MD simulation with the help of GrowMax 2020.4 up to 500 nanoseconds on HPC cluster. So as earlier Sir Patil sir said about it, and also protein ligand, then protein peptide, then protein simulations. And also next one is nothing but PBSA kind of calculation, particularly these calculations for the binding free energy. And that PBSA is nothing but molecular mechanics, Poisson-Boltzmann surface area. And actually that method we are going to use to calculate binding free energy, particularly for protein ligand complexes. And also it can be used in the study of biomolecular kind of interactions. The next one is the PCA analysis. That PCA analysis also, that is nothing but principal component analysis. And that is one kind of, we can say, statistical procedure. And that statistical procedure, particularly to summarize the information content, and that content in a large data table. Okay, so by means of we can do it in the smallest set of a summary. That is nothing but PCA kind of analysis. The next one, cluster analysis, and last one is nothing but Desmond-based MD, molecular dynamics simulation. This is up to also 500 nanoseconds, and actually that Desmond kind of MD simulation that computes both the absolute as well as the relative solvation free energies. That is nothing but regarding with this Desmond kind of MD simulations. This is all about regarding with the synthetic chemistry and computational chemistry. Then we are also going to provide the facilities from analytical chemistry also, which includes the analytical method development and validation, particularly for the API as well as the formulations also, which includes the dissolution and all this. And then stability and degradation studies of the various kinds of pharmaceuticals also. And also some of the extraction part, that is from the pharmacognosy department. That is nothing but we can provide the phytochemical or we can say chemical constituent isolation from the various kinds of plants with the help of different chromatographic technique, which includes the column chromatography and other techniques also, SFE also we can use for that. Then next one is nothing but phytopharmacological screening, that is particularly from pharmacology department, and standardization of the herbal drugs also with the help of various spectroscopic methods, which includes the UV, IR, HPLC, HPTLC, all these we can provide such kind of facilities. And also, as I earlier said, that is nothing but isolation and the characterization of the various kinds of chemical constituents from the natural products, and also quantification of the API in dosage forms with the help of spectroscopic techniques, which includes UV, IR, HPLC, and HPTLC also. And also in addition, we can also provide the facility regarding with the other chromatographic techniques, which includes the flash chromatography and the column chromatography. So all these facilities we can provide from the pharmaceutical chemistry department. So thank you very much. And now I would like to request Yogyata Pathare ma'am to explore the areas from pharmaceutics department. Thank you.
Very good afternoon to all the respected dignitaries, respected principals, sir, and faculties, as well as my dear students. So herewith, I just give a brief introduction of the Sutics department. I'm the assistant professor in Sutics department, and herewith we are representing the various research facilities which we are having and various research projects which we already completed. So these are the various core research area where we are working. Most of them we are already completed in the form of an PG project as well as in the form of an PhD project. So here, just it is with respect to the formulation and development of an oral dosage form, as the many of the conventional dosage form having a problem that we have to take a medicine again and again, and that's why there is a toxicity problem. Also, there is a less patients compliance. So we are developing a various sustained release and immediate release tablets so that we can minimize the dosing frequency as well as we can maximize the efficiency of the drug. Then we are also going for the bilayer tablets where we can prevent the various incompatibility problem, and also we can maintain the dose, we can maintain the concentration of the drug in a plasma within the therapeutic window. Then in a new area, we are working in a mouth dissolving tablets where we require a maximum absorption through the mouth and we are getting a fast action of the drug. Then gastroretentive drug delivery system, due to the GI transit time, most of the drugs are get washed out before getting fully absorbed. So here we are developing a various gastroretentive drug delivery system like the floating drug delivery system. Then fast disintegrating oral films. Many of the patients, they are having an difficulty in swallowing the tablets. So we are also develop a different types of an oral films which are disintegrate within 30 seconds in the mouth itself. So these are with respect to the various oral dosage form. Then this is our major core research area, and this is the nanoparticulate drug delivery system. So by using this nanoparticulate drug delivery system, we are also develop the oral as well as an ophthalmic as well as an transdermal and nasal products. And here we are incorporated in various nanoparticulate systems. So here just a diagrammatic representation of the different nanoparticulate drug delivery system which we are developed in these research activities. So majorly we are going for the nanoparticulate system like in solid lipid nanoparticles, the gold nanoparticles, liposome, micelles, and nanosphere. And this is also the one of the area where the PhD work is going on for this area. So here, as the many ophthalmic products, they are having an what we can say that disadvantage that the drug cannot stay for longer period of time, and that's why we have to be administered or we have to be instill the dosage form or eye drop 3 to 4 times a day, and that's why this will become a toxic to the delegated eye tissue. So we are developing here the various nanoparticulate system like an ophthalmic nano emulsion, ophthalmic nano suspension. Also, we are going for these nanoparticulate dispersed in situ gel, where when in a second diagram where when we instill it, it is in the form of a liquid, but when it is come in a contact with the lacrimal fluid, it is get converted into the gel, and that's why we can increase their residence time. And in the third here, we are working so in the form of an ophthalmic film, so that we can just place the ophthalmic film and we can get a drug release up to 7 days also, because most of the marketed preparations are also available in this area. Then formulation and development of a transdermal drug delivery system. So this is also the one of the major core area where the tremendous research is going on at our college itself. So here we are preparing the various transdermal patches, and now also our PhD students are developing the dermal assisted types of a model like an iontophoresis, so that the drug are get majorly absorbed through the skin. So this is just a representation of this transdermal drug delivery system. These are the other areas of pharmaceutical research. As you know that majority of the polymers are of an synthetic, and that's why it is not compatible to our body and it is responsible for the toxicity. So here we are concentrating on the natural polymer and we are developing the natural polymer so that it can be used for the pharmaceutical use in the form of an binder, in the form of an viscosity enhancing agent and so on. So here also we are working currently, we are working on the different natural polymer like and tamarind gum is there, rosin is there, and various cellulose alginate derivatives are there. So that we are in the process of developing so that if we can use it is in the formulation. Then this is also the one of the part that is of solubility enhancement methods by using the cyclodextrin and other method because most of the drug are poorly soluble. So there is a need to enhance the solubility and then we can go for the their dosage form. So we are also going for such a type of methods. Then we are also we are also developing the in vitro models for the drug testing. So currently in last year, we have developed the transcorneal model in order to check the corneal permeability of the drug. Also similarly, we can also go for the transnasal as well as transdermal. And then test masking of a drug. As you know that we don't like to take a drug because it is very bitter test. And so that's why we are also working on this test masking of the drug. So these are the list of the research project. Many of the research projects are already completed and some of them are in a pipeline and maybe completed within these few years. So these are the list of the research projects which we carried out at our research center. These are the main instruments which we are having via which we are conducting such a type of research activities like the nanoparticle size analyzer. We are having a dissolution tester. We are having a tablet compression machine, tablet coating machine, then diffusion testing apparatus. Since we are formulating the ophthalmic and such a type of sterile products, so we require a class 100 area and class 10 area. So we are also having a class 100 area, laminar airflow unit, stability chamber, etc. So these are only a glimpses of the some of the important instrument which we are going or which we are using for carried out such a type of research activity. And this is the pictures of one of our pharmaceutics lab, that is of an industrial pharmacy lab. Totally, we are having a 6 pharmaceutics lab under the heading of microbiology, physical pharmacy. Also, there a dissolution testing lab is also there. So this is one of just a glimpses of the industrial pharmacy lab where the maximum instrument we are there and students are performing their research activities here. So this is a short introduction, or you may say that the presentation of my department. So thank you so much. Now I request Mahadik Madam to present the cognoscity part.
Thank you, Yogita Madam. Good afternoon to everyone. I'm here to represent the pharmacognosy department. And as we know, this pharmacognosy is a basic lead or is a basic crucial department which is interlinking for because it is providing the link or the leads for the research for pharmaceutics, for chemistry, as well as for the pharmacology as well. So as we are having the PhD department for the chemistry and the sutics, but this is dealing with the crude drug. So it's upcoming at the developing stage, even though it is at the developing stage, we are right now having certain facilities and we are regularly performing certain activities which will be helpful for research work at a very basic level. Because as we know, the pharmacognosy is dealing with the crude drugs and the identification of the right drug is very important for the research work. And obviously, we at the pharmacognosy department are dealing with the drug evaluation by performing the very basic stage, right from the morphological identification, that is nothing but the external evaluation or the organoleptic evaluation of the crude drugs, which can be confirmed by performing the various microscopic tests by taking various transverse section. As we know, as everyone, we are having the our fingerprint, but our fingerprints are identical to us. Similarly, the whatever the cellular structure the drugs are having, that is their identity of that cellular arrangement of the crude drugs. And we at our department, we are having the regular practicals, or we are performing regularly the activities of performing the transfer section. So we are helping out here to do the microscopic as well as the basic morphological evaluation of the drug. Then After that, as we know, the crude drugs are the combinations of the various phytochemicals. So, a number of the phytochemicals are present in each and every part of the crude drugs. So, we are here to do the analysis from the basic stage, like performing the proximate analysis, and by performing the various physiochemical properties, say right from starting the moisture content, ash values, extractive values, bulk densities, tap density, angle of repose, and various other parameters which are very helpful for the evaluating the drug for its quality. So not only the quality, but the quantity of the right amount of the quantity of the phytochemicals is very important in each and every part of the plant. So by performing some quantification determination, we are here to helping out or to assist for the determination of the quantitative determination by performing the various parameters here, like loss on drying, ash value, acid insoluble ash values, and water soluble ash value, and many more. So apart from that, powder characteristic is also the important part for the identification of the drug. We are also here to assist for doing the powder characteristic of the crude drugs. Then after that, many of the as I told you, the phytoconstituents are present in the plant, but unless until they are isolated from the plant, we cannot use that. So for isolation of these constituent, we are here to provide the different extract performing the various extraction process, right from the conventional extraction process like infusion, decoction, digestion, maceration, etc. And the Soxylate hot extraction method, which is a very common method and which is important method for the separation or preparation of the extraction. Apart from that, we can also provide the extraction of the phytoconstituent by the ultrasound with the help of ultrasonic extraction and the microwave assisted extraction, which will help us for increasing the yield of the phytoconstituent, and whatever the conventional methods, they are time consuming methods. So depending upon the need of the phytoconstituent, to reduce the time of the processing and also to increase the yield, these two processes, advanced techniques we are having here, and we can give the assistance by providing these methods of the extraction. Similarly, whatever the extraction provided, if you are having your extracts and you want to analyze that extract for the further identification of the constituent, we can provide the analysis just by starting from the preliminary phytochemical investigation up to the advanced analysis of the phytoconstituent. And apart from that, isolation, purification, and detection of the active principles also we can provide by with the help of various spectroscopic and the chromatographic techniques, as my colleague Dr. Sadhna ma'am explained in her session. So apart from that, many of the phytoconstituent they are having, or many of the crude drug, they are having their activity because of the presence of volatile oil or the essential oil. So it is required to be isolated from the plant part, and as they are thermolabile, or they, as the name indicate, they are essence or they are volatilized in the nature, they require certain different techniques for the isolation. So right from the conventional method of the using hydro distillation apparatus or the Clavenger apparatus, in addition to that, we can provide the ultrasound and microwave assisted hydrolyzation for this isolation of these volatile oil as well. So these are certain diagrammatic representation in the down portion. There is a combination of ultrasound which increases the surface area of the crude drug, and which will reduce the time further processing the isolation of the volatile oil. So apart from that, if you are having your herbal formulation, say for example like Asava and Arista, we are very much here to help you to determine the alcohol content, because it is a very important part of this formulation to be analyzed. So we are here to determine the alcohol content of such formulations like Asava, Arista. Then we are also dealing with the evaluation of the excipients which we are going to use for the herbal formulation by performing various parameters. Then preparation of the herbal cosmetic formulations. If you want assistance, we can give you the assistance for the preparations of the herbal formulations like cream, lotions, shampoos, and many more. Then preparations of the herbal formulations like syrup, mixtures, tablets. We are we are performing those formulations. Apart from, if you want the assistance for the confirmation or the to study the monograph of the drugs, so with the help of the reputed pharmacopoeia, official pharmacopoeias, we are here to provide the assistance for the monographic analysis of the crude drug. Then some of the people, they may want the analysis to be done for the phytochemical or the chemical constituent present in the formulations like aldehyde, phenol, alkaloids, which are the secondary metabolites and important for the various activities. So we regularly perform this activity at our department. So these are the whatever the facilities we are having these in the pharmacognosy department, which is the first primary step of the herbal drug technology. So with this, I conclude my presentation, and I request Divya Thita madam to continue.
Good afternoon, one and all. I am Divya Thite, Assistant Professor of Siagat College of Pharmacy. Today I am going to introduce about the pharmacology department. So there are pharmacology department, there are various sections like acute toxicity study, reproductive study, mutagenicity study, also ecotoxicity study. And this first one is facility and research capabilities. In this facility and research capability study, operating the platform of offering high quality research service and the highly good laboratory practice experience scientists, also center for the drug discovery and the development. And they perform the research with facility comprising of a custom design experimental from in silico study to the clinical studies. In this next one is there are various toxicity study are carried out. First one is acute oral study in rat. It is turnout month in 1 month. It is second one is acute oral study in rabbit in turnout in 1 month. Also carried out the study dermal study in rat, acute inhalation study in rat, acute dermal irritation, corrosion, and the study in rabbit, also mice and rat. Also guinea pig maximization test in sensitization. In this guinea pig maximization test sensitization also carried out the corrosion test. In this next one is subacute studies and the reproductive chronic study. In this subacute studies, first one is a oral study in rat. In this turnout month with 0.6 month. In the second one, subacute dermal study in rat, also subacute dermal study in mice as well as rabbit. In this guideline OECD 4010, and this turnout the month is 0.3 month. The subacute inhalation study in the rat for the 28 days. And the second one is reproductive chronic study. First one is teratogenicity in the rat. Also teratogenicity study are carried out chicken eye, and the second generation study in the rat in the turnout month is 10. Third one is carcinogenic study in combined toxicity. The turnout month is 32 month. Next one is the genotoxicity study. In the genotoxicity, the amylase subluna micro some assays. In this turnout month is 0.1 month. Second one is chromosome aberration in CHO cell lines. OECD guideline for 473. Also in few micronucleus test in mouse. Next one is chromosome aberration test in mouse. And this last one is genotoxicity study is in vitro micronucleus study test in mouse. Next one is ecotoxicity study. In this ecotoxicity studies, freshwater algae growth inhibition test and this immunization test in diapnea species, also acute toxicity to fish, zebrafish, and this acute toxicity to fish, chicken, vegan, and the reproduction in birds like chicken, acute toxicity to earthworm, honeybees, and the zebrafish. And the, in pharmacology department, other study are carried out in neurotoxicity study, also cytotoxicity study, human health monitoring study, and this neurotoxicity also, as well as hepatotoxicity study and the nephrotoxicity. In this next one, let's stop in vitro model studies. First one is in vitro cytotoxicity study. In this MTT method. Second one is mammalian cell gene mutation test, cell transformation assay, also vitro anti-diabetic assay, glutoactivity based anti-diabetic drug screening, and the hepatoprotective activity. And there are a different animal disease model with talks in lab also undergo the sponsored design animal model, and some UG and PG student are carried out this activity anti-asthmatic studies, anti-diabetic model in rat, also mice, anti-inflammatory test in mouse, anti-tussive activity in guinea pigs, anti-ulcer activity in rat, anti-pectic activity, anti-stress activity, tumor activity, hepatoprotective, into the CCL4 induced model, as well as the alcohol induced model, and this hyperlipidemic activity, inflammatory bowel disease in rat and mouse, and the wound healing activity. This lab facility of the pharmacology department. So I will request to Ms. Amruta ma'am to give the brief introduction to today's guest speaker, Dr. Ramdas Kute sir. Thank you.
एक्झिट करून तिथून करावं लागतं. व्हॉल्यूम कमी कुठून कमी केलं?
Thank you, ma'am. Good afternoon, all present over here. It's my immense pleasure to introduce today's guest speaker. Thank you, ma'am. Good afternoon, all present over here. It's my immense pleasure to introduce today's guest speaker. Dr. Ramdas Kute sir. Sir has completed graduation in Ayurved BMS from Pune University in 1997. In 2005, he started career as a general practitioner, inception in Indu Pharma and Ayurvedic Medicine Manufacturing Company in Jejuri MIDC. In 2016, Indu Pharma expanded as Inducare Pharma Private Limited, engaged in manufacturing of Ayurvedic medicines, herbal cosmetics, nutraceuticals, and food products. With the motto of think pure, eat pure, and use pure, the developed products are exported to all over India and in other countries. Sir currently involved in joint business delegation as a representative of pharmaceutical industry with Honorable President of India, Mr. Pranav Mukherjee, and Prime Minister Narendra Modi. And he also appointed as Managing Director of Inducare Pharmaceuticals and Research Foundation, SPV, assisted by Department of Pharmaceuticals. Government of India erecting first common facility center for R&D and common testing laboratory and common logistics center with grant in addition of 20 crore out of total project cost 50 crore. Sir is president of Jejuri Industrial Manufacturers Association Jejuri and Kadepata Rural Medical and Research Foundation Jejuri, also Shatayu Hospital and Ayurvedic Research Center Jejuri. Lastly, Hindu English School Jejuri, and he also editor of Ayurved Ayurved Vichar, ISBN and ISSN, registered quarterly magazine dedicated for healthcare. With this brief introduction, I request sir to proceed with the session. Thank you.
Good evening, everybody. Hope everyone is ready to listen me after a heavy lunch. I think it's 4 p.m. now. First, I am thankful to Honorable Principal Sir, Dr. Rajesh Patil Sir, and Dr. Badhe Sir for inviting me for a noble cause. I congratulate and wish all the best and happy birthday wishes to our principal, Dr. Rajesh Patil Sir. So in the evening, everybody can ask him about it. I congratulate all the team accompanied by Dr. Sadhna Raut ma'am, Yogyata Pathare ma'am, Mahadik ma'am, and Divya ma'am. Under the guidance of Dr. Praveen Badhe for this inaugural session of Center for Drug Discovery and Development. You might, everyone knows the story of hare and tortoise. मला वाटतं आपल्या सगळ्यांना सशा आणि कासवाची गोष्ट माहिती आहे. तुम्ही सगळेजण हो म्हणताय, परंतु मला खात्री आहे you don't know the full story. What Dr. Rajesh Patil sir has told you in his addressing lecture. Everyone knows here challenges to tortoise for a race. Here was speedy but not consistent, and so the tortoise win the race. Everyone knows the story, but nobody knows what happened next. So ego of here challenges tortoise once again for the race. And after ego, you know everyone that no doubt here will win the race. What happens next? Again, the ego of tortoise challenges the hare for one more race. But tortoise keep one condition, that the route will be decided by me. And of course, the route was passing through the river. You know who will win the race. Story doesn't end here. Again, here, ego of here challenges tortoise once more for the race. You know, again, here will win the race if the race route was not selected by the tortoise or but the river. Both did sit with each other and they decide why we are fighting with each other. Because tortoise can cross the river, here not. But on the road, here will be more faster than tortoise. and both they have sit under a tree where here taken a rest in the first race, and they decided why will we will not support each other. In ancient era, there is a one nyay that is the andha pangu nyay. So whenever a race starts, here says tortoise, come on my back. He ran faster. Whenever a river comes, tortoise said, here come on my back, and they will cross the river. So the race is completed within a record time with the support of limitations of here and support of limitations of the tortoise. The same thing, what Dr. Rajesh Patil sir has done in your institute today, that different departments like chemistry, biology, cognosy and statistics, they may have their own limitations, but he, with the support of Dr. Badhe sir, he has given a credit to Dr. Badhe sir, that with the support of and under the guidance of Dr. Badhe sir, he brought all the 4 departments under one roof. Might be they are competitors before today in number of respect, number of activities. कधी कधी होतं की सुटिक्सच्या माणसाचा डान्स गॅदरिंग मध्ये चांगला होतो, कोलोजीवाले नाराज होतात, कोग्नोसीवाले कधी कधी असं म्हणतात की नाही आम्ही ट्रॅडिशनल लोक आहोत ना. So what our principal sir has done, that he learned from the here and tortoise. He learned from the history like अंधपंगू न्याय. आपल्याला ती पण गोष्ट माहित असेल, रस्त्याने जायचं असतंय, आंधळ्याला रस्ता माहित नसतो पण चालता येतं, आणि पंगू म्हणजे पांगळ्याला चालता येत नाही म्हणून रस्ता माहित असतो. मग काय होतं, पांगळा आंधळ्याच्या खांद्यावरती बसतो आणि दोघं मजेत रस्ता क्रॉस करतात. मला वाटतं whatever medicines we are manufacturing, whatever medicines we are developing, it requires the symbiotic efforts of chemistry, of cognosy, of pseutics and biology. So I congratulate Dr. Rajesh Patil sir. He took the opportunity to start such a novel center on his birthday and World Hepatitis Day. So I congratulate him. Sir also expressed about the DDD. In our school, lot of people, everyone knows who see the Hindi movie, JD, JK, DD. It might be the same thing, DDD, but I think this DDD will definitely overcome the deficiency of vitamin D of the researchers. So you can take the concept of C and DDD like this, that no department will be deficient with the support of other department of the research projects. Sir has described about the research support for agriculture science. Being a graduate of Ayurveda, you have gone through my CV. And for the first line, you might know, आज किस गधे को पकड़ के लाइए, तो आयुर्वेद का आदमी है. So basically, I am from Ayurveda science, so I don't know about the pharmacy. I don't know about the pharmacology. I don't know about the pseutics and all the hectic and tricky branches which we think. I am little bit close to cognosy because of herbal food drugs. But ethnopharmacology or reverse pharmaceutical engineering is nothing but a branch where Ayurveda can touch it. As Madik Madam, I think Madik Madam has rightly expressed her support to the department through the Department of Pharmacognosy for the research purposes of crude drugs or herbal drugs. I would like to request your CNDDD team to consider our faculty, that is Ayurveda, which will support you and which will help you for the upgradation of the center, and it may open one of the very good branch for the research. As Mahadik Madam has rightly expressed, that through the organoleptic study, she can identify the drug. In extension with that, I will add one more thing, that in Ayurveda, there is one principle that is a Purushayama Loka Samhita. The meaning of that a human is a zip copy of this universe. So everyone carries pen drive with him or her, which contains all the pharmacy, right from the undergraduation to post-graduation. The same thing happens. Human being is nothing but a zip copy of a universe. So everything in universe is a replica, whatever is in human being. The same principle will be utilized when you think of a juglans regia, that is accord. If you see the organoleptic, if you see the morphology of the juglans regia, then it is nothing but a brain. And now modern scientists had came to know that it contains omega acids, which nourishes the brain. So this principle is nothing but the basic idea to study the cognosy department and to help for the formulation and basically for the pharmaceutical chemistry people, how we can go for the reverse pharmaceuticals. Do you know first antihypertensive? Can anybody know the first antihypertensive? That is serpina. It is extracted from the Rolfia serpentina. It is the world antihypertensive. Even Madik madam had described that before 1990s, almost all the medicines derived from the plant extracts. And that's why if a human being, if his diet is considered is nothing but a mostly all the material which is available in the universe, of course, to treat the disease, all the materials, right from the soil up to the gold, everyone is needed either to keep his health maintained or to treat the disease. So pharmacognosy branch can be upgraded with the support of Ayurveda, and I request to the department that please consider such a person or may take advice of these people which will help you number of things. As I have given one small example of Juglans regia. Patil sir expressed expertise of Mali sir about the genomics and next generation sequencing. I think he is more competent in that field. It is useful as far as the prevalence of cancer or oncology is concerned. And with the support of biotechnology, with the support of nanotechnology, we must can find out solutions to treat the different types of cancers. Patil sir has given example about the vaccines and availability of the vaccines of vitamin C. Sorry, vaccines of hepatitis C. The same thing happens about the oncology department also. Because though the word cancer contains only 5 English alphabets, it is a huge branch and the world is striving for that. And of course, gold nanoparticles or nanotechnology definitely will target the cells of cancer, which are similar to the body, which are similar availability, but requires a specific target. And I think once again, in Ayurveda, concept of bhasma is closely related with the nanoparticles. As chemistry department has extended the support to the researchers, to the students, and to the pharmaceutical companies. This chemistry department has a huge role and a huge scope as far as elemental or molecular chemistry is concerned. And in Ayurveda, we use number of metals. मराठीमध्ये आम्ही तर असं म्हणतो की मातीपासून सोनं, सोन्यापर्यंत आणि हिऱ्यापासून प्लॅटिनम पर्यंत प्रत्येक घटक वापरणारे आम्ही आयुर्वेदाचे माणसं आहोत. But when we study, when you go for the analysis, there is a huge gap between what our ancient scientists expect and what we are providing to the society. And that gap will be bridged by the chemistry department with the research or with the support of elements and molecules. So transpermeability or transcellular activity of a molecules, either it may be gold nanoparticles, you can make a silver nanoparticles, you can make a different types of nanoparticles, which is included in Ayurveda, what we call is as a bhasma. So Department of Chemistry may have huge scope in that area. While discussion with Patil sir, as in our introduction, organizer has described about our upcoming project. Sir, I would like to inform all the researchers on whom shoulders responsibility of healthcare is there. We are introducing ICP-MS, we are introducing LC-MS-MS, we are introducing GC-MS-MS, we are introducing UV spectrophotometer, and along with that FTIR also. Because it is the need of 21st century. Use of chemical compounds in your day-to-day life. As you know, whatever food you are eating, it may contain preservative. Whatever utensils you are using, either they are herbal, they are natural, so-called, without chemicals or without molecules, it can't be used. And so now developed world like US or European Union are very much cautious about the contaminations. We are thinking about the PPB level, but now US FDA is thinking about the PPT level, particles per trillions. So this will be one of the newer door opening for the researchers, especially in the food articles like fresh foods, because number of food like fruits and all these things, they may contain large quantity of pesticides. So certification of organic products or certifications of pesticide residue free products is mandatory for the export. And so one more branch is opening, that is analytical chemistry. And definitely I'm confident about your chemistry department that they will take care of them, apart from the identification, synthesis, and isolation of the molecules in a routine day-to-day life. Sir has described about the development of vaccine. Everyone know the importance of vaccine after COVID pandemic, outbreak of COVID pandemic. In the 20th century, vaccine had changed the life of human being. मला माहित नाहीये की आपल्यातले कित्येक लोक आजी आजोबांच्या संपर्कात असतात, पण ते सांगतात की आमच्या काळामध्ये मर्यायचा गाडा येत होता, आमच्या काळामध्ये देवी यायची, आमच्या काळामध्ये पटकी यायची. या सगळ्या गोष्टी आपण वी हॅव डिमॉलिश, वी हॅव almost finished, including polio, only because of vaccines. And vaccines had changed the era of the pharmaceutical industry. And you know, everyone have a dream to join serum only because of the glamour of the serum. Every pharmacy students or postgraduate student always think, Bhagwan ko kuch mangna hai to serum mein job mango. So vaccine is one more area where your team can work. While discussing with Bade sir and Mari sir, I came to know that you are fortunate, you people and your organization, your institute very much fortunate, being you are Srihangar Technical Education Society's institute. Because see, once we start with unique, or you may travel with alone, nothing can be done. But when you will travel with your organizations like Srihangar Medical College, Srihangar Dental College, I think biotechnology is also part of your organization. So, as rightly explained by Bade sir, that you people are in a view of a collaboration with your other sister concerns, which will bring a lot of new ideas, including artificial intelligence and machine learning, which can be just, you can hire from your electronics department just close to you. So being a student or being a faculty of a Srihangar group, it will be very easy to collaborate with each other for the betterment of the society. And definitely it is a good thing for Srihangar College and this newly developing center also. You might know, everyone know the statistics that when we isolate 10,000 molecule, only one molecule reaches to the consumer or to the patient. Because there is a huge line of the selection of molecule, selection of target molecule, then synthesis, isolation, and then clinical trials might be from the phase 0 to 4, and then FDA approval, and then market of that product. It's a very tricky job. And everyone knows India is a hub for pharmacy, world pharmacy. We are the second largest pharma product exporter after China. But remember that we are exporting 99% of the drug which are out of patent. Unfortunately, there is a label on India that we are a copy master. Though we are providing economical pharmaceutical products to the world, what we do, we are very much expert in processing and isolation of a molecule, those who are out of patent. India is exporting almost all the molecules which are out of patent. So there is a huge scope in the discovery of a molecule, but it's a very hard job. Everyone knows that it takes years right from the target selection to the commercialization of the product. And about the finance or about the about the funding required for that is a huge. But institutes like CNGAT, who can take a support from your big hospitals, which will be utilized for your phase 2, phase 3, phase 4 trials. You can work as a clinical research organization with the support of your other sister concerns. And definitely, as Patil sir have shown you one dream that something positive will definitely come out from this institute because of your competitive collaborative efforts. And that is very much important. I have seen the presentations of the Sutix and came to the number of formulations has been developed, like control release, drug release, sustained release, fast release. I would like to congratulate, I think, Pathare ma'am, who has expressed in a very lucrative manner. I request her to add one more new concept, that is effervescent tablets. I think she might have done work on that, but not in her presentation. But paracetamol in effervescent, number of multivitamins in effervescent is the developed market is going very fast. And as everyone knows, effervescent tablet needs a dry granulation, and that granulation and compression that is in a very specialized area. It has to take care of relative humidity, temperature also. So it must be processed in a clean room with HVAC or conditions. We are manufacturing number of effervescent tablets, and definitely your department will work on that, because now bilayer tablet, entry coated tablet became an old fashion. But now newer and newer goods are coming. And so effervescent in the form of tablet is one more important thing. Madam has expressed about the transdermal route application of the products. In Ayurveda, we uses oils and ghee, which is a precursor of, these are medicated oils. And I would like to add one speciality about that, that most of the medicated oil preparation, we use milk. In Sanskrit, it is called as Godugdha. We add milk because of enhancement of the absorption, because absorption of milk is more faster than absorption of water percutaneous. In ancient Ayurveda, when a patient or when a child suffers from diarrhea and dysentery, unable to take a sip of water, it is mentioned that just massage with milk. He will need not to give any antibiotics or any rehydrating solutions like ORS. This is the magic because skin absorbs milk fast. And as all most of the females know, all the skin applicants, right from the fairness cream to any lotion, they are always making the advertisement that it contains 25% milk. एक बंदा तो बोलता है 75% milk ही है उसमें. This is not true. It's but everyone claiming that it contains milk. So synergistically, such a traditional medicine manufacturing people can work with you, especially for the formulations, and that will resolve the issue of artificial polymers also. There are huge scope, especially in analgesic or painkiller or drug pain relieving agents because of side effects. A lot of people are still relying on paracetamol. If people from last 20 years, there are a huge development right from the nimesulide to etarocoxib, celecoxib. Number of coxib came into the market, but because of huge side effect, all these coxibs have been vanished from the market, and still people are relying on the traditional medicines, either like paracetamol, ibuprofen, or diclofenac sodium or potassium. But now complete developed world is aware about the side effects of painkiller or analgesics, and they are thinking about the local application formula or local applicant medicine, which will work to relieve the pain. So that area is also very important, and you people can work on that. I have seen the laboratory, and I congratulate right from the founder of this institute to the principal and complete faculty, because you can do a lot, especially in cognosy. Lot of postgraduate students and researchers, they require chromatographic study, or they require fingerprinting, which can this facility can be availed from your cognosy department. Lot of developments can be done, especially in the formulation department, and definitely this center will help you. We all people are working for the healthcare of the society. When I was in the cabin of Honorable Principal, sir, I saw one sentence that we are not merely pharmacists. We are not here to develop one pharmacist, but we are taking care of the person, that he must be a human being also. So in the same way, you people don't have to think like that we are a pharmacy graduate, either postgraduate, we will have a career. But 21st century is the huge scope for pharmacy people, especially in research. And now Government of India has focused on the research. You all people know that we say Jai Jawan, Jai Kisan. Now this government has added Jai Jawan, Jai Kisan, Jai Vignan, and Jai Anusandhan. That is, research is a very important thing. What we need, again, as our principal sir said, that copy is very easy. And up today, we have done a lot of copies. What we do, we have copied all antiretrovirals, and we are giving in 1 rupee per day. As you know, HIV medicines given by CIPLA is 1 rupee. Why? So we have done a lot of copies. Now we have to be focused on research, and definitely such institutes and newly inaugurated center will open the door for the research and to be have some contribution, special contribution in the human being. But what we need, again, the center and our principal said rightly, that is DDD, that is dedication, determination, and devotion. And we have to forget about the mobiles and about the WhatsApps and Facebooks, because a lot of people still searching and chatting. So this is very important, because without, as like Patil sir has expressed, that Badre sir had joined in January, and in July he has opened one center. So I will say big applause for him also, because this is what dedication, determination, and devotion. Being a newcomer, such center opening or inauguration of center is not possible without dedication, devotion, and determination, and without the support of our staff, without the support of our principal, and without the support of our organization. So once again, I wish you all the best to all your students who will be part of this center. Something will definitely come out, because 10,000 people, 10,000 young male and females are visiting Mumbai to be a star, but only one becomes a star. 10,000 people, more than 10,000 people are thinking to become a MLA, to become a MP, but only one becomes. The same thing happens. 10,000 molecules will be taken out, but only one will reach to hand to the consumer or to the customer to get relief from his disease. And definitely it will be from the C100. Thanks once again.
Thank you, sir. Thank you for your inspirational words. Now, Dr. Rajiv sir is with us. I will request Dr. Rajiv sir to say a few words for Lakshmi. Okay, Dr. Lakshmi ma'am is with us. I will request Dr. Lakshmi ma'am to say a few words for today's program.
Hello. Are you able to hear me?
Yes, yes. Your voice is coming, ma'am. Just 1 second.
Yeah.
Yes, ma'am.
Am I audible?
Yes, ma'am, you are audible.
Okay. Shall I go ahead?
Yeah, yeah.
Okay. Good afternoon to everyone. Thanks very much for the invitation. It was enlightening to see that such centers. Are they able to hear me? Are they able to hear you? Sorry, hear me? Hello?
Hello. Yes, ma'am. You can. Yes, we can hear you, but there is echo. So just few seconds.
Yeah.
Ma'am, you can start, ma'am.
Yeah. Okay. Thank you. Good evening, everyone. Thanks for the invitation and having me here. It's pleasure and it's actually very enlightening to see a center that is opened in undergraduate college like this, which is really a good platform for the students. Um, so I mean, the principal sir was pointing out a lot of areas, and it's all a wonderful opportunity to for the students. And actually, one thing which I like to point out, the students, the undergrad and the postgraduate students are very young minds, and they'll be very enthusiastic about and excited about a lot of things, and that is the best time to start these basic research and get go on board. I'm very happy. I think this center will be a good platform and good place for collaboration. Hope soon we will also come with my course there, and hope to, whatever the way that technology aspects that CCAM, Center for Cellular and Molecular Platforms, or my expertise will help the students, I will be really happy to contribute. Thanks very much, Dr. Praveen, for invitation and taking forward and giving an opportunity to uh, introduce my course also soon. Hope we will work soon with that as well. Thanks very much.
Dr. Gayatri Deshmukh ma'am and Priyanka Kumar, who all has spare time from his busy schedule to grace the occasion. Today, we have an opportunity to hear your thoughts, and this will surely be going to encourage us in our future events. My heart fills with lots of gratitude for our today's guest. They enlighten us with their comfortable talk on the today's topic. Now, they talk on a various aspect related to the research and enhancing our understanding towards this topic. Thank you once again, sir and madam, for your valuable guidance to all. We are thankful to our Honorable President, Professor M.N. Navle, sir, Founder Secretary, Dr. Mrs. Sunanda Navle, Madam, Vice President Admin, Mrs. Rachna Navle, Ashtekar, Madam, Vice President HR, Mr. Rohit M. Navle, sir, for their motivation. I'm running short. Words to express my humble thanks to our principal, sir, Dr. R.B. Patil, sir, for arranging, planning, and making this meeting successful. Your support and motivation lead us on the path of success. I would like to thank Mr. Praveen Bade, sir, and our teaching and non-teaching staff for their active participation and also for presentation in making this session successful. Once again, thank you. And one and all just contributing this meeting, I conclude this session. Now, I request everyone to please gather in front for photo session.